Oxidative stress, autophagy, and cellular senescence form a deeply interwoven triad at the heart of aging and cancer biology. Each process influences the others in subtle, yet profound ways, and maintaining their balance is essential to preserving cellular function and longevity. At the center of this relationship is autophagy, the body's internal waste disposal and recycling system, which plays a critical role in neutralizing oxidative damage and preventing both premature aging and malignant transformation. Reactive oxygen species, ROS, are natural byproducts of cellular metabolism, especially from mitochondrial activity. While small amounts of ROS serve important signaling functions, excessive ROS generation overwhelms the cell's antioxidant defenses and leads to oxidative damage of DNA, proteins, and lipids. This accumulation of damage is one of the hallmarks of aging and a major driver of cancer. Damaged mitochondria, in particular, become a vicious source of further ROS, creating a self-amplifying cycle of destruction if not properly managed. Autophagy serves as a crucial protective mechanism against this type of stress. Through a specialized form known as mitophagy, cells identify and remove dysfunctional mitochondria before they can leak excessive ROS. This containment prevents the spread of oxidative damage and preserves mitochondrial quality, which is vital for energy production and cellular homeostasis. When autophagy is functioning properly, ROS levels are kept in check, and the cell remains in a state of dynamic equilibrium, constantly renewing itself and avoiding the triggers of senescence or malignancy. As we age, however, autophagic capacity declines. Lysosomes become less efficient, autophagosome formation slows, and key regulators like AMPK and sirtuins are downregulated. As a result, damaged mitochondria begin to accumulate and ROS levels rise. This unchecked oxidative stress contributes directly to the onset of cellular senescence, a state where cells cease dividing but do not die. These senescent cells secrete pro-inflammatory and pro-growth factors, creating a toxic tissue environment known as the senescence-associated secretory phenotype, SASP. This environment not only promotes aging and tissue dysfunction, but can also encourage nearby precancerous cells to progress toward malignancy. The collapse of autophagy in this setting thus removes a vital layer of protection. Without efficient clearance of ROS and senescent cells, tissues become inflamed, energy depleted, and vulnerable to transformation. This cascade illustrates a dangerous feedback loop. Oxidative stress impairs autophagy. Impaired autophagy fuels further oxidative stress, and together they push cells towards senescence or cancer. The warning here is stark. When the balance between damage and repair tips too far, the body enters a trajectory of decline that becomes increasingly difficult to reverse. Yet this very connection also offers a powerful therapeutic opportunity. Restoring autophagy, even partially, may be one of the most effective ways to reduce oxidative burden, clear senescent cells, and extend cellular health. Lifestyle interventions such as intermittent fasting, exercise, and plant-based diets rich in antioxidants have been shown to reduce ROS levels and stimulate autophagic activity. Pharmacological agents like rapamycin, metformin, spermidine, and NAD+ boosters are under investigation for their ability to mimic these effects, essentially rebooting the cell's cleaning systems and breaking the cycle of stress and damage. There are, however, caveats. In some disease contexts, stimulating autophagy could inadvertently help damaged or precancerous cells survive. ROS is not only a source of damage, but also a signal for apoptosis in defective cells. If autophagy suppresses ROS too effectively in the wrong context, it might prevent the natural elimination of cells that should die. This highlights the importance of context. Age, tissue type, disease state, and genetic background all influence whether boosting autophagy will heal or harm. Despite this complexity, the outlook remains promising. Emerging evidence suggests that carefully timed interventions, especially in midlife before extensive senescent burden develops, may offer the greatest benefits. By maintaining autophagy at optimal levels, it may be possible to prevent the accumulation of ROS, delay the onset of cellular senescence, and dramatically reduce the long-term risk of cancer and age-related decline. In the grander picture of aging biology, autophagy appears less like a single pathway and more like a conductor of cellular harmony, coordinating stress responses, metabolic adaptation, and repair. When it falters, oxidative chaos ensues. But when preserved or restored, it offers a path back to equilibrium, a biological checkpoint with the potential to halt the spiral of aging and disease. As science moves closer to targeted strategies for regulating autophagy, oxidative stress, and senescence, we edge toward interventions that don't just treat the symptoms of aging but address its underlying mechanics at the cellular level.